Hello dear learners, I am Dr. Sabiha Wali, ex-professor in Food and Nutrition and Head Postgraduate Department of Home Science, Rashtrasant Tukdoji Maharaj, Nagpur University, Nagpur. Welcome to this module on anti-nutritional factors. Dear learners, in this module, I shall be giving you a brief outlook of the nutrients present in foods based on chemical structure, biosynthetic origin and specific action they exhibit in the human body. The module will also cover simple processing methods to inactivate the inhibitors. The detailed description of each of these inhibitors will be discussed in the upcoming modules. Now, let us see what are the objectives of this module. After studying this module, the learner will be able to describe anti-nutritional compounds with their occurrence, classify anti-nutritional factors, understand the biochemical effects of anti-nutritional factors, and finally discuss the methods of processing. Now, let us see how anti-nutritional factors are defined. Anti-nutritional factors are defined as those biological compounds that are present in human or animal foods and that reduce nutrient utilization or food intake, and thereby they contribute to impaired gastrointestinal function as well as metabolism. Now, food components that deliver positive physiological functions to humans are termed bioactive compounds, while anti-nutritional compounds refer to those food substances that negatively interfere with the digestion as well as absorption of nutrients. Now, learners, this classification is however debatable, since currently some of these compounds could offer both beneficial as well as adverse effects. Now, the majority of biochemical compounds and anti-nutritional compounds are secondary metabolites. These are produced primarily by plants as a defense mechanism against invasive predators and or environmental stress. Plants commonly synthesize a range of secondary metabolites as part of their protection against attack by herbivores, insects and pathogens, and this is a means for plants to survive in adverse growing conditions. Now, anti-nutrition compounds are also termed as defense metabolites, having specific biological effects depending upon the structure of specific compounds, which range from high molecular weight proteins to very simple amino acids and oligosaccharides. Now, anti-nutrient substances from nutrition point of view interfere with normal growth, reproduction as well as health when consumed regularly in amount existing even in normal component of diet, and therefore they should be considered harmful as well as toxic. Now, these secondary plant metabolites cause depression in growth and performance and health due to a variety of mechanisms, including reducing protein digestibility, damaging the intestinal wall, thereby lowering digestive efficiency. Now, anti-nutritive compounds can interfere with food components before intake, during digestion in the gastrointestinal tract, as well as after absorption in the body, thereby affecting the functioning and utilization of nutrients and may lead to nutritional deficiencies. There are several heat stable and heat labile anti-nutritional toxic and potentially toxic factors. The anti-nutritional factors mainly occur in pulses and grains, legumes and foods, and feed material prepared from grain, legumes and pulses. It is worth noting that while there are many groups of chemical compounds that have health benefits, others can be very toxic as well as fatal to humans when consumed. The list is inexhaustible and therefore it warrants concern. Now learners, let us see how anti-nutritional factors are classified. Now these anti-nutritional factors in plants may be classified on the basis of the chemical structure, the specific actions they bring about, or their biosynthetic origin. On the basis of chemical structure and heat stability, anti-nutrients are grouped as number 1 proteins and number 2 other substances. Now proteins include lectins and protease inhibitors. These are known to be sensitive to normal processing temperatures. Other substances which are stable or resistant to these temperatures and which include among many others polyphenolic compounds, mainly condensed tannins, non-proteins, amino acids and galactomannans gums. The other classification, let us see how anti-nutritional factors are classified in other ways also. There is a broader classification which categorizes anti-nutritional factors into the following 3 types. Type A, these are substances primarily interfering with the digestion of protein or the absorption and utilization of amino acids. Antiproteins, for example, are protease inhibitors, lectins and hemagglutinins. Now type B, these are substances interfering with the absorption or metabolic utilization of minerals. Now antiminerals like phytic acid, oxalic acid, dietary fiber and gossypol. The type C anti-nutritional factor, an antivitamin or vitamin antagonist is a substance that makes a vitamin ineffective. It lessens or negates the chemical action of a vitamin in the body. These are a group of naturally occurring substances which can decompose vitamins from unabsorbable complexes with them or interfere with their digestive or metabolic utilization. This module, learners will throw light on the adverse antagonistic action of anti-nutrients based on the latter classification. Now under type A, anti-nutrients that we discussed before are protease inhibitors and lectins. Now let us see what are protease inhibitors. Protease inhibitors are the most commonly encountered anti-nutritional factors of plant origin and are widely distributed within the plant kingdom, including the seeds of most cultivated legumes and cereals. Now trypsin and chymotrypsin inhibitors are present in raw legume seeds. They have the ability to inhibit the activity of proteolytic enzymes within the gastrointestinal tract. Trypsin inhibitors are found in raw soybean and they inhibit the protease enzymes in the digestive tract by forming indigestible complexes with dietary protein. These complexes are indigestible even in the presence of high amounts of digestive enzymes, thereby affecting protein digestion adversely. They, however, decompose with heat. When legumes are consumed raw or without being cooked properly, they upset the digestive tract. Now coming to lectins, these are carbohydrate binding proteins present in most plants, especially in seeds like cereals, legumes and certain oil seeds including soya bean, in tubers like potatoes and also in animals. They function as both allergens as well as hemagglutinins and are present in small amounts in 30% of foods, more so in a whole grain diet, and hence ingested daily in appreciable amounts by both humans and animals. Most hemagglutinins of higher plants are found in seeds, but tubers and plant saps are also sources of lectins. Lectins are most commonly found in leguminous seeds. These are characterized by the ability to combine with carbohydrate membrane receptors. One of the most important feature of plant lectins is their ability to survive digestion by the gastrointestinal tract of consumers. Now, in such a situation, it allows the lectin to bind to membrane glycosal group of cells lining the digestive tract. As a result of this interaction, a series of harmful local and systemic reactions are triggered, placing this class of molecules as anti-nutritive and or toxic substance. Now, locally, lectins can affect the turnover and the loss of gut epithelial cells, damage the epithelium, interfere with the nutrient digestion and absorption, stimulate shifts in the bacterial flora and modulate the immune state of the digestive tract. Systematically, they can disrupt lipid, carbohydrate and protein metabolism, promote enlargement and atrophy of key internal organs and tissues, and alter the hormonal and immunological status. Now, when lectins are consumed in excess by sensitive individuals, they can cause three primary reactions. Number one, severe intestinal damage, disrupting digestion and causing nutrient deficiencies. Number two, provoke immunoglobulin G. Now, immunoglobulin G is an antibody that is found in all body fluids, as well as immunoglobulin M. This is an antibody which is found mainly in the blood and the lymph fluid, causing food allergies and other immune responses, and can bind to erythrocytes. Simultaneously, the third one is they bind to erythrocytes simultaneously with immune factors, causing hemagglutination, resulting in anemia. Now, to come to the conclusion of this model, I hope learners, you have understood the terms used, that is bioactive compounds and anti-nutritional factors. Food components that deliver positive physiological functions to humans are termed as bioactive compounds, while chemical substances interfering with the proteins, vitamins and minerals and reducing their availability in the body are termed as anti-nutritional factors. Now, these are also known as secondary metabolites, as discussed earlier, because they act as a defense mechanism against insects and pathogens for the plants. Now, anti-nutrients have been classified in different ways. We have seen that they have been classified based on chemical structure or their specific action, and these classification of anti-nutritional factors based on specific action has been discussed in this module as type A, that is anti-proteins, type B anti-minerals, and type C antivitamins. Now, type A category of anti-nutrients, that is protein inhibitors and lectins, and their role in interfering with the digestion and absorption of nutrients were discussed in this particular model.